Can't believe anything's fun in biology. It is. It's kind of the fun stuff. Unfortunately, today is a lot about vocabulary. But once you learn the words, you're okay. Okay? So we're going to do genetics. This is often referred to as classic Mendelian genetics. And then after we do classic Mendelian genetics, we'll take it a step further and we'll talk about non-Mendelian genetics. And it's usually a lot of fun. So Gregor Mendel, however you want to say it, Greg, he is responsible for the laws that govern the inheritance of traits. He's often referred to as the father of genetics. So we'll just call him Dad for short. So this is in 1822. He lived from 1822 to 1884. So if we take a look at him, he was an Austrian monk. You know where Austria is? Miss Rivera is Austrian, not Australia. So he was an Austrian monk, and he studied the inheritance of traits in pea plants. Like the peas that you eat. And the reason that he did that was because you could grow a lot of plants really quickly. Each one of the little peas that they make would be considered an offspring or a baby. And you could grow them in a relatively small area. And through his work with pea plants, he developed the laws of inheritance that we still use today. However, they kind of just looked at him as like this crazy guy working in his garden. It's like, oh, don't mind Gregor. He's out in the garden again. Because they kind of thought he was a little bit crazy because his work wasn't really recognized as being valuable until about the turn of the 20th century, which would be about the 1950s. Why do you think that was? Not in the 50s? Yeah, technology was swell. What happened in the 1950s? Perfect. Discovery of DNA. Thanks to those thieving gentlemen, Watson and Crick. So based on the idea that they were now saying, yes, DNA was the genetic material, all of the stuff that Gregor Mendel had said about like these invisible units being passed on from parent to offspring suddenly made sense. So he died not knowing that he had been successful. He did his work between 1856 and 1863, and during this time he cultivated, meaning he grew, and he tested or observed some 28,000 pea plants. It's a ton of pea plants. And he found that the plant's offspring retained the traits of the parents. Now, that to you guys is like common knowledge. Yes, children look like their parents, usually. But in his time, it was kind of revolutionary. And that's why we refer to him as the father of genetics. Look how happy he looks there. Not really. Now, if you would like to see where it is, there right here, where this little plot of flowers is planted, that's where Gregor Mendel actually had his garden. It's no longer in Austria. It's now part of the current Czech Republic. And you'll never really have to know that, but unless it's on HQ. So current day Czech Republic is where you can find the plot of land that Gregor Mendel used to grow all of his pea plants. The idea, or the ideas that Gregor Mendel, we've referred to it as particulate inheritance, because he said that physical traits, give me an example of a trait. Hair color, perfect. So these physical characteristics are going to be inherited some way as particles. He didn't know what they were. He just knew that these little particles that are passed on from parent to children make them look similar. But he didn't know that the particles were actually chromosomes and DNA. I mean, remember, it wasn't until the 1950s that they finally conceded to the fact that we as humans, have DNA as our genetic material, not proteins, even though we're super complicated. So here's some terms that you need to know. The first term is a trait. And Tripp, you gave me a great example of a trait. What is a trait? Give me an example again. Tripp, give me another one. Oh, unattached or attached earlobes. That's one of my favorites to look at. Who has attached earlobes? Oh yeah, you do have really attached earlobes. By definition, is any characteristic that can be passed on from parent to offspring. So it's going to be any characteristic that can be passed on from parent to offspring. Tongue rolling, that's another one. If you can roll your tongue.
I caught myself.
I am not I can't do it. I'm not coordinated enough, I guess, to teach myself. But that's, and tongue folding, that's another one. Oh, it's so gross. Stop. Double jointed, well, there's some other things that go into that. So, for example, if you have a connective tissue disorder, you might be double jointed, and that's not necessarily something that has to be passed on from parent to offspring. It could be caused by a random mutation. When we say heredity, what do we mean by heredity? It's just the passing on of traits. So passing on of traits, so those characteristics from parent to offspring. Well then, isn't that the same as genetics? No, because genetics is the study of heredity. So the traits are the characteristics, the heredity is the actual passing on of the characteristic, and genetics is studying how those characteristics are passed on, or that process. So they're three words, very similar, maybe used synonymously in the vernacular, but in class they have very specific definitions. We're also going to talk about both monohybrid and dihybrid crosses. I'm sure you guys all remember how to do it. We're going to look at the magic little graph that we use to help solve genetic problems on the next slide. Mono means what? One. A hybrid, if something's hybrid, we're like, what's a hybrid car? I know Prius, but gas and electric, right? So it's a combination of things. So a monohybrid cross is going to be a cross between two organisms involving a single trait. So a single characteristic. So an example, like for Gregor Mendel and his magic pea plants, one of them could be so an example would be something like plant height only. Or looking at flower color or seed color. Just one thing. What do you think dihybrid means? What is di? What you'd like to do in biology? No.
Two or more.
Not two or more. It's just two. So it's a cross between two organisms, or so a cross between two organisms involving two traits. So an example with this would be like wanting to know if you're going to have a tall plant and yellow flowers. What is the probability? I want tall plant, yellow flowers. So both of them at the same time. So two different traits at the same time. So maybe like brown hair and brown eyes. Well, how do we figure that out? Well, the only way to actually determine what the offspring would look like is to actually make them mate. We're not doing that in here.
I have a question.
Yeah. Yeah.
For the example you said height.
So that would be just looking at what's the likelihood of having a tall plant?
Oh.
Or what's the likelihood of having a yellow flower. A dihybrid is I want to know what the likelihood of having a tall plant with yellow flowers, so together, okay? So to do that, we use a Punnett square, and the Punnett square is used to help us give the probability of offspring in genetics problems. So the Punnett square, it's going to be used to show the probability of offspring. Does it show the actual offspring? No, the only way to do that is to do a cross. For example, the likelihood of having a male child and a female child in each pregnancy is always 50%. So that means if I had 4 children, I should have 2 girls and 2 boys. And it actually did work out that way for me, but it was just like a total fluke of, a fluke. There are some families where they have like 5 kids and they have all boys, or 5 kids and they have all girls. So the Punnett square is only going to show us the probability of having offspring. The only way to actually have the offspring is through going through with the actual mating. So we're going to go through, is there anyone in here who has not made Punnett squares? Because if you've all done this before, then I don't need to go on.
I might have, but I don't remember.
Okay. So on the outside here, you have the female and the male gametes. What's a gamete? Sex cell. What's the female gamete called? Egg. What's the male gamete? Sperm. So for each trait, you have 2, we're going to look at these terms called alleles. And you got one from mom, one from dad. And so on the outside, we're going to separate them into the possible eggs and sperm you can make. And then in the middle here, they combine to make the possible offspring. So on the outside, you show the gametes, and on the inside, it shows the possible offspring. We like to say on the outside, it shows the process of meiosis, which is making your sex cells, and on the inside, it's the process of fertilization. So how would I make a Punnett square? Well, the easiest way to do it, Charlie, you're going to make a grid. All right, step number 1. I made my grid. Then what you're going to do is you're going to put the alleles for gametes on the outside. So for here, I'm going to put the female. I'm going to say big T and a little t. And here I'm going to make the male, big T. Oh, it's a little male too. And a little t. And then on the inside, once you have your gametes selected, and we'll go over how to select gametes in a second, then what you do is you combine the alleles in the boxes. So if I combine them here, I'd have one big T, one big T. On the next one, what would I have, Charlie?
I can't tell.
That's a big T and a little t. And then this one here, I'd have 2 little t's and 1 big T and 1 big T from each one. That's how you would fill out your Punnett square. Well, what is that going to tell you? Well, these letters are going to correspond to a trait, and you're going to use that to be able to determine what they would look like and what their genotype and phenotypes would be. So let's do some more vocab, unfortunately. You have alleles. I keep using this term allele. What is an allele? We use letters usually, right?
Yeah, 2 letters.
But they're not really letters. What do they really represent?
The traits.
Well, you're close. It's going to be the 2 forms of a gene. Right? And those 2 forms of a gene are either going to be dominant or recessive. What does it mean if something's dominant? Right, so the dominant, I like to think of it as I'm the dominant and you're the recessives. Haha. So when I'm in the classroom, I overshadow you. If I were to leave, the room would come alive with the voices of all the recessives, right? So the dominant is often referred to as the stronger of the two genes. You only need one dominant allele to have a dominant trait. And how do we usually represent a dominant trait or a dominant allele? Very good. And we usually show it with a capital letter. Now, I want to tell you that just because something's dominant doesn't mean it happens more often. Who knows what polydactyly is? Poly means what? Many. Dactyl refers to digits. Polydactyly is having more than 5 fingers or toes. That's a dominant trait. How many of you have 6 fingers? Oh, but wait, it's dominant. It should happen more often, right? No. Dominant just means you need one allele to have that trait. It doesn't mean it happens more often. Well, then what's the recessive? Well, the recessive is the one that is, it's the allele that can be masked by the dominant. So it only manifests in the absence of a dominant allele. So, and that's why in this case you need 2 recessive alleles to have a recessive trait. How do we usually show that? The lowercase letter. Very good. When I look at you, can I say, oh, you've got 2 dominant alleles? No. What do we see? We see how they look. I can't see what letters you have, right? I can't see your alleles. I see the trait. So that means we have 2 different ways we can talk about these traits. We can talk about the genotype and then the phenotype. The genotype is the allelic combination. How can I say that more simply? Well, how about we just say it's the letters? So your genotype, for example, this could be big S, big S, big S, little s, little s, little s. Those are the allelic combinations for this specific trait. So the allelic combination for a trait would be the genotype. The phenotype is the physical manifestation resulting from the genotype. So how can I say that easily? How about how it looks? Right? So for example, the phenotypes in these could be smooth, it could be smooth, or it could be wrinkled. So the phenotypes would be smooth, that's how it looks. The genotypes for something that's smooth could be big S, or big S, little s. So let's give an example. So if we look at a flower, let's say that I write big R is equal to red, little r is equal to yellow. What can you tell me about that?
Red is dominant, yellow is recessive.
How did you know that? Capital letter, very good. So he said red is dominant, yellow is recessive. Notice that it's big R and little r, and it's not R and Y. You want to try and use the same letter to represent the trait. So absolutely, so red is dominant to yellow. We know that all genes occur in pairs, right? Because you got one from mom and one from dad. So that means you need 2 alleles for each trait. So what are the possible ways that we can mix up big R and little r? Well, you can't just say RR. They're all going to be RR. You have to be a little more specific. I sound like a seal. So I could have big R. And then what else could I have? Big R, little r. And then I can also have? Well, that's the same thing. Little r. If it's big R, what are these things that you've just told me? Big R, little r. What are those going to be? Genotypes or phenotypes? Genotypes. What would be the phenotypes for big R? Red flower, blue thorn. How about big R, little r? What would that look like?
Red.
But wait, it's got a little r. I thought, wouldn't it be orange?
It dominates it.
Oh, it's dominant, so I don't show it. Very good, because I only need 1 allele for the dominant trait to manifest, so it would also be red. What about little r? Yellow. Very good. Well, we have special terms we use when something's going to have the same letters or the same alleles or different, 2 different alleles. So they could be either homozygous or heterozygous. What do you think homozygous means? The same? Give me an example from our last one with the flowers. Which ones would be homozygous? Trip. They're all RR. Big R, and little r. Very good. So homozygous, the homozygous genotype, this is going to be the genotype where your combination involves either 2 dominant or 2 recessive alleles. So they have 2 of the same alleles. So in this case, it would be big R, or little r. Another term for this would be purebred, or pure breeding, or true breeding. So pure breeding, true breeding, they mean they're homozygous for a trait. What do you think heterozygous means? Well, what does hetero mean? Hetero is different. So this would be where the gene combination or your allelic combination contains 1 dominant and 1 recessive allele. We also refer to this as a hybrid. Pretty easy, right? It's a lot of words.